Hello guys, this is Kendall Terry, and today is a discussion of DNA, specifically the research that went into discovering that DNA was the genetic information, what it looks like, and how it replicates in essence. And that's what we're primarily going to be focusing on the slides today. The first is looking at 1928, Frederick Griffith, and Frederick Griffith did a simple experiment. He took live virulent Streptococcus pneumoniae. Now, live virulent means that it was a bacteria that was alive, and virulent means disease-causing. So he puts these, injects them into a mouse or a rat, and I'm sorry, these are like mutant, weird-looking rats, but it's the best I can do. So you inject that into this rat, and the rat dies. Then you take live non-virulent Streptococcus pneumoniae. So now you've got live non-virulent, meaning not disease-causing, injects those into the rats live. Then he heat kills virulent bacteria, so now the bacteria are still the virulent ones, the ones over here, but they're heat killed, and injects them into the mouse or rat, and the rat lives. Then he takes heat killed virulent, so these guys right here, the rat lives, and live non-virulent, these guys right here, the rat lives, mixes them together, injects them into the rat dies. And what Griffith said in this was that apparently the live non-virulent bacteria were transformed by picking up something from the virulent ones. And he called this transformation. So then the question was, well, what was causing the rat to be the bacteria to be transformed? Was it protein or was it DNA? That's the question. So there was some more experimentation that had to be done. So we get to 1944, Avery, McLeod, and McCarty, and their research all kind of comes into this understanding that transformation was due to DNA, not proteins. And this is some of the things that they did. They found out that the elemental composition agreed closely with that of DNA. So that's the first thing. Whatever's being transformed looks very closely to what we know DNA is. Extracting lipids and proteins did not reduce transforming. So you take lipids and proteins away, transformation still taking place. Protein digesting enzymes did not affect transforming activity, nor did DNA digesting enzymes. RNA digesting enzymes. So you take protein and RNA digesting enzymes, put those into the mix, bacteria can still be transformed. When you put DNA digesting enzymes into the mix, all of a sudden it destroyed all transforming activity. So no transformation takes place anymore now that DNA is being digested. So that would go to support that what's actually going on during transformation is DNA is being picked up by one bacteria from the other, and they're incorporating that into who they are, and then that means that they express the stuff that they've picked up on that DNA. In the case with Frederick Griffith, that would have been the ability to be virulent to the rat or kill it. So now we have Hershey and Chase. And Hershey and Chase did their experiment in 1952. You can see we're progressing. They took sulfur, radioactive sulfur, and grew bacteriophage in radioactive sulfur. Bacteriophage is a virus that affects bacteria. That's what it's hosted. Now, the thing about the bacteriophage is that all of this on the outside is protein. This little bit on the inside is DNA. So they grow this bacteriophage in radioactive sulfur. The reason is because sulfur is a very major part of proteins. So if there's radioactive sulfur where they're being grown, then the proteins will be radioactive, but the DNA for the most part is not going to be affected. Then they allowed that bacteriophage to infect bacteria. Then they would blend and centrifuge the cultures, and what they found was that the radioactive sulfur was actually out here in the liquid, not in the bacteria. So it's not the sulfur or the protein was actually being left out here on the outside. It wasn't being injected into the bacteria. So if the protein's not being injected, then the protein can't be the genetic material that has the code, because whatever's being injected into the bacteria would have to have the code on it that would cause more viruses to be made. So then they took radioactive phosphorus, 32P, and they grew bacteriophage in that culture. Now in DNA, one of the main components is a phosphate group, more so than in protein. So you're going to have now radioactive phosphorus incorporated into DNA, but not radioactive phosphorus really found in the protein. So now they infect bacteria, they blend it, centrifuge it. Now, when you get over here to the culture, you find that the phosphorus, the radioactive phosphorus, is actually found down here in the bacteria pellet at the bottom, saying that the radioactive material has been injected into the bacteria. And that would support that DNA is what is carrying the genetic code, not proteins. That's a big disclaimer that has to be made, because even in 1953, when we get over to the next slide, when Watson and Crick did their big discovery, that was still one of the questions. They thought it was DNA, but it was still that toss-up between DNA and proteins. That was still going on. So let's look at DNA a little bit here and see some of the structures. DNA is made up of a nucleotide. DNA and RNA are both nucleic acids, but it's made up of a nucleotide. A nucleotide is a phosphate, sugar, and nitrogen base. RNA is also made up of a phosphate, sugar, and nitrogen base. The difference between these two has to do with right here. DNA has just a hydrogen. RNA actually has a hydroxyl group. So this is deoxyribose nucleic acid, and this is ribose nucleic acid because it has that oxygen right there. And these nitrogen bases are one of 5 nitrogen bases, depending on which nucleotide you're looking at. Each of them actually only have 4 possibilities, but there's a switch down here that takes place. There are 2 categories, purines and pyrimidines. Purines, there's 2 types. There's adenine and guanine. They're 2-ringed, meaning kind of like this is a ring right here. There's 2 of those put together. And the way I remember them is Ag on the periodic table is silver. So if I'm getting silver from someone, I want it to be pure silver. Purines are Ag. In the pyrimidines, we've got cytosine, uracil, and thymine. In DNA, it's cytosine and thymine. In RNA, it's cytosine and uracil. Okay, there's a little switch there. These are 1-ring structures. There's only 1 ring like this in these structures. And the way I remember these is I always think about cutting the pie. If I've got pie there, I want to cut it, right? So CUT, cytosine, uracil, thymine, are pyrimidines. All right, that's information that you need to know so that we can go on to this next step here. So now we've got these 3 that all kind of come together to figure out not only that DNA was a genetic code, but what it looked like and how it worked. So Chargaff was the first one, and through Chargaff's research, we found that the amount of adenine present in DNA always equals the amount of thymine. He also found the same to be true for guanine and cytosine. So he said no matter how much adenine I have, there's always the equal amount of thymine. That's an interesting combination. It's an interesting discovery. Franklin and Wilkins then go on to look at X-ray diffraction. Now part of their research looked at DNA, but that was not their main research. They were actually doing some other things. But X-ray diffraction analyzed DNA in the shape of a helix or corkscrew. When they looked at this X-ray diffraction, which is kind of like an X-ray picture of molecules, when they looked at it, it had the same kind of shape that a helix has of other things that they know are helical, what they look like. So you would say, well, DNA is probably then in some kind of shape of a helix or corkscrew. Now, many up to Watson and Crick in 1953 were thinking of a 3-stranded with like a center backbone and 3 strands on the outside of it. But whenever they would try to build that in the cell itself, in the environment that the cell's in the nucleus, it wasn't possible. The chemistry just didn't support it. So Watson and Crick, using this information and other information that they had, they were able to build a model of DNA as a double helix with A binding to T and C binding to G to make like the steps of the double helix. Whenever this got put together, one, it showed us what the code was. It showed us that it was an easy system. If A's on this side, T has to match here. If C's here, G has to match here. Okay? And that's a system that can then be replicated down the line because it's not very complex. Most of the things we find in nature have a simple mechanism about them. DNA, simple mechanism once we figured it out. So if DNA has that simple mechanism, then how does DNA replicate? Okay, there's several different setups here that could have been possible. Some said, well, you've got a DNA and parts of it will break apart and other parts will fill in. Some said that it'll just look at the strand itself and build a new one over here. But the one that actually comes out to be is here, which they call the semi-conservative method. What they said is, Messelson and Stahl in 1958 found out that the DNA would literally split apart and you would have a new strand, or an old strand on each side, and then a new strand would actually fill in on each of the old strands, and you would end up with two strands of DNA, one with a new strand and an old strand, over here, a new strand and old strand. The way they figured that out was they took E. coli and they grew it in 15 nitrogen medium. That is a heavy nitrogen. Then they transferred that E. coli to a 14 nitrogen medium. That's a lighter nitrogen. So they took some E. coli straight out of that before they allowed it to grow, and they centrifuged it, and they found the DNA was down here in the bottom, which is where the heavy nitrogen is going to be. They took DNA 20 minutes after inserting it into this, which would allowed it to go through one round. That E. coli would have been one round of binary fission or splitting, so the DNA would have had to have been copied one time. And they found that the DNA was actually made up of a mix between the light nitrogen and the heavy nitrogen. Then they allowed it to go 40 minutes, 2 rounds, meaning 2 times the bacteria have divided at this point. And when they centrifuged it, they found there was DNA that was a mix of both heavy and light, and then there was DNA of light. The only way for this to happen is for that semi-conservative method, for you to have the old strands separate, new strand, and that gives you then your DNA. Then, as that split again, you would end up still with that original old strand of heavy nitrogen, new strand of light nitrogen, new strands of light nitrogen would bind to it. So you would end up with some of that still, a little bit of both, and then the others of completely light nitrogen DNA.